In the previous subtopic, we have learned about that photosynthesis consists of two stages. First stage is light reaction because they require light energy for a few processes, and followed by the second stage, which is the Kelvin cycle, which they do not use light energy, but they use the product produced from light-dependent reaction. And today we are going to focus on the first stage of photosynthesis, which is light-dependent reaction. Synthesis occurs in the organelle chloroplast, and light-dependent reaction occurs at the thylakoid membrane, or the membrane of thylakoid, and they absorb the light energy of photon by the photosynthetic pigments, or chlorophylls. The absorbed energy will be converted to chemical energy such as ATP and NADPH or H+. In cellular respiration, we learned about that the high energy molecules such as NADH and FADH2 will be produced. But in photosynthesis, the high energy molecule involve the NADPH. That's the P here. And at the same time, at the end of light-dependent reaction, oxygen molecule will be produced as a byproduct. There are two types of light-dependent reaction. The first, non-cyclic electron flow, involves the Photosystem 2 or P680 and Photosystem 1 P700. The name non-cyclic implies that the electron will flow from one origin or the electron donor, and then they will go to the last electron acceptor, which is a different component. However, cyclic electron flow consists of photosystem 1 only, or P700. They show us how the electron can be transferred from one component to the next component, and at the end, it will return to the origin. It means that the electron donor and the last electron acceptor are the same component. The electron flow is important in the process called as photophosphorylation. In cellular respiration, we have learned about this term. Phosphorylation is something related with synthesis of ATP. So, in light-dependent reaction, photophosphorylation is the synthesis of ATP that excited by light energy, or photon. This is why they have the prefix photo. And if we refer to the ATP produced by the non-cyclic electron flow, this is a method called as non-cyclic photophosphorylation. But if the ATP produced is due to the cyclic electron flow, this is a method called as cyclic photophosphorylation. So sebenarnya ATP dihasilkan melalui proses fotosintesis lebih kurang sama dengan cellular respiration, iaitu semasa elektron bergerak dari satu komponen kepada komponen seterusnya, tenaga itu akan dibebaskan disebabkan perbezaan tenaga antara dua komponen tersebut, dan proses redox reaction mesti terlibat. Before we move on to the details about the light-dependent reaction, we have to recall the components of photosystem. And there are two types of photosystem, which are PS1 and PS2. Each photosystem consists of reaction center, and the reaction center consists of primary pigment, which is usually found in a pair. And the example of primary pigments are chlorophyll A P680 and chlorophyll A P700. Different types of the pigments depends on the photosystem. Is it either PS1 or PS2? And in the reaction center, there is another component called as primary electron acceptor. Primary means that they are the first electron acceptor before they pass through the electron transport chain. Other than that, we have a few accessory pigments surround the photosystem, which include the other examples of chlorophyll A and chlorophyll B and carotenoid. Now let's correlate the structure of one photosystem with this process, photoactivation, which is a process triggered by absorption of light energy or photon. So first, light energy or photon is absorbed by accessory pigments, and they're able to transfer the pigment from one accessory to another one until they reach at the primary pigment found in the reaction center. When the energy absorbed by the primary pigment, the electron in the primary pigment is increased to a higher energy level and photoexcited. The photoexcited electron is accepted by primary electron acceptor. This is called as photoactivation. And what happened to the reaction center is they come with electron deficiency because the electron will keep leaving from the primary pigment to the primary electron acceptor. Other than photoactivation, another function of light energy or photon is it able to stimulate photolysis of water. It means that it lyses or split the water molecule by light energy or photon. So here is the equation of photolysis of water. One water molecule can be split into oxygen atom, 2 protons and 2 electrons, with the absorption of photon at chlorophyll or bichlorophyll, which occurs at the thylakoid membrane. If we look at the product of photolysis one by one, the oxygen atom will combine with another oxygen atom to form oxygen gas, which is considered as a byproduct, iaitu hasil sampingan, and the oxygen will be released into air. However, the 2 protons or the 2 hydrogen ions will be released into thylakoid space. The last is the 2 electrons. It will be used to replace the photoexcited electron from the PS2 or the P680, which means it able to overcome the electron deficiency occurs in the photosystem. Let's study the non-cyclic electron flow, or how the non-cyclic electron flow contribute to non-cyclic photophosphorylation. It begins with the absorption of light energy or photon by the accessory pigments of PS2, because the non-cyclic photophosphorylation begins with the photosystem 2, and the photon is transferred from one accessory pigment to another one until they reach at the primary pigments in the reaction center. Because this is PS2, so the primary pigments is actually the chlorophyll A P680. So yang penting jangan keliru dengan anak panah ini. Ini sebenarnya membawa masuk pergerakan atau transfer of the tenaga. Sebab ramai senior akan anggap ini adalah elektron, tetapi sebelum sampai di reaction center, semua anak panah yang melibatkan accessory pigment adalah tenaga sahaja. So when the energy is absorbed by the primary pigment in the reaction center, the electrons in the primary pigments can be increased to a higher energy level until it becomes photoexcited and accepted by the primary electron acceptor. So here is the occurrence of photoactivation. Photoactivation will bring the problems called as electron deficiency, and it can be overcome by the photolysis of water. Because each water molecule can be splitted by the light energy or the photon into two electrons, and these two electrons can replace the lost photoexcited electrons from the primary pigments. So the P680 molecule in the PS2 now return to the reduced state. It becomes stable again. And at the same time, from the photolysis of water, it releases two protons and oxygen atom, which is going to become oxygen gas and released as a byproduct. So, when the photoexcited electrons are accepted by the primary electron acceptor, it can cause it become very unstable, and primary electron acceptor will pass the two electrons to the next component found in the ETC, which is the plastoquinone. So during the passing of electrons from primary electron acceptor to the plastoquinone, it involves redox reactions because the primary electron acceptor is oxidized and the plastoquinone is reduced. When a component accepts the electrons, it will become a high energy level. So immediately they will pass the electrons to the next component, which is the cytochrome complex. And redox reaction occurs again, and this process repeated during the electron flow. The ETC or the electron transport chain consists of plastoquinone, or abbreviation PQ, cytochrome complex, which is actually a proton pump, and plastocyanine, abbreviation PC. So if we study this diagram carefully, we notice that the energy level is decreasing from plastoquinone to plastocyanine. So when the electrons are passed from plastoquinone to the cytochrome complex, energy will be released. Same goes to the passing of electrons from cytochrome complex to plastocyanine. They will release the energy. And the released energy will be used to activate the proton pump, which is the cytochrome complex, to pump the proton from stroma into the thylakoid space. It means that the proton will be pumped from low hydrogen ion concentration at the stroma to the thylakoid space, which has the higher hydrogen ion concentration. And this will create a proton gradient between these two spaces until chemiosmosis able to occur. So the concept same as we studied in the cellular respiration. When chemiosmosis occurs, it activates the enzyme ATP synthase to combine ATP with inorganic phosphate group to produce the high energy ATP. Okay, class, we have to understand that photosystem is found in the thylakoid membrane, and thylakoid membrane is just a very small part that can be found in the chloroplast. So if photosystem 2, they absorb the light energy or the photon at the accessory pigment, that means at the same time the photosystem 1 will be able to absorb the light energy of photon by their accessory pigments also. And the absorbed photon or the light energy will be transferred from one accessory pigment to another until they reach at the primary pigment at the reaction center. And for the PS1 or the photosystem 1, the primary pigment is actually the chlorophyll A P700. And the electrons in the primary pigments will be increased to a high energy level until they become photoexcited and accepted by the primary electron acceptor. So photoactivation occurs at PS1. Photoactivation occurs at the reaction center of PS1 or photosystem 1 will cause electron deficiency. But this problem can be solved by the photoexcited electron that release from the PS2 and transported by the electron transport chain until they reach at the P700. And this will help the P700 molecule in PS1 return to its reduced state. The photoexcited electron released by P700 in PS1 will be accepted by primary electron acceptor. And primary electron acceptor is reduced, so they will pass the electrons to the second electron transport chain, which is ferredoxin. And the ferredoxin is reduced, so redox reaction occurs. And the ferredoxin will pass the two electrons to the last electron acceptor, which is NADP+, which will combine with the two protons and become NADPH with the balance of one proton. This reaction is catalyzed by the enzyme NADP+ reductase. So this is a picture to show us how the relation between the photosystem 1 with the second electron transport chain, which is the ferredoxin or abbreviation FD, and the enzyme NADP+ reductase located at the membrane of the thylakoid. So when the NADP+, the last electron acceptor, bind at the active site of this enzyme, they will accept the two electrons from the ferredoxin and combine with the proton from stroma and produce high energy molecules, which is NADPH. And this NADPH will be released into the stroma for the next activity. So this picture summarizes how the product can be formed from non-cyclic photophosphorylation. So we start with the first product, which is ATP. ATP is formed because of the electron flow happens in the first electron transport chain. So during the electron flow, the energy will be released and it's able to activate the proton pump, which is the cytochrome complex. They pump the proton from the stroma into the thylakoid space. And this pumping of proton will cause the saturation of hydrogen ion in the thylakoid space, or the proton gradients are generated between the membrane of the thylakoid space and the stroma until chemiosmosis occurs at the ATP synthase, which catalyzes the formation of ATP molecule in the stroma. The second product is high energy molecule NADPH, which is formed at the stroma also, whereas the NADP+ will accept the electron from ferredoxin and combine with the proton. These two products, ATP and NADPH, are going to be used in the next reaction, which is the light-independent reaction called as Calvin cycle. Now look at the second type of photophosphorylation, which is cyclic photophosphorylation that closely related with cyclic electron flow. And cyclic photophosphorylations only involve PS1, photosystem 1, where the light energy of photon can be absorbed by the accessory pigments of PS1. and the photon is transferred from one accessory pigment to another until they reach at the chlorophyll A P700, or which is actually the primary pigment in the reaction center of the PS1. And the electron in the PS1 will be increased to a higher energy level until it becomes photoexcited. The photoexcited electron will be accepted by primary electron acceptor. This is called as photoactivation. So same as before, if photoactivation occurs, it leads to a problem, which is electron deficiency. The photoexcited electron released from the primary pigment of PS1 will be accepted by the primary electron acceptor. And when the primary electron acceptor is reduced, it will pass the electrons to the first component of ETC, which is ferredoxine. So redox reaction occurs. And the electron will pass to the next component, which is the cytochrome complex, and the last is the plastocyanin. So the energy level is decreasing from the ferredoxine to the plastocyanin. So during the transfer of electrons within the ETC, they will release excess energy, and the energy will be used to activate the proton pump, which is the cytochrome complex. And this will pump the proton from the stroma into the thylakoid space. So from the low concentration stroma to the high concentration thylakoid space, they are able to produce a proton gradient, which means the proton will diffuse back to the stroma through ATP synthase, and the process is called as chemiosmosis to produce the ATP molecule. But the photoexcited electron that happens in the primary pigments of reaction center in PS1 will cause electron deficiency, and this will be replaced by the electron from the plastocyanin. Okay, this is why we notice that the P700 molecule in PS1 will be returned to the stable state, which is the reduced state, because they accept the electrons. And if we study this diagram one more time, this is why they are given a name of cyclic, because the electrons is flow in a cycle. If compared with non-cyclic photophosphorylation, this shorter pathway only produces ATP, because the electron do not pass from the ferredoxine to the NADP+. They will pass from the ferredoxine to the cytochrome complex and to the plastocyanin, and returns back to the P700 in PS1. In plants, both non-cyclic photophosphorylation and the cyclic photophosphorylation are observable. However, the non-cyclic photophosphorylation are more common. And the cyclic photophosphorylation occurs when the Kelvin cycle or the light-independent reaction, they require more ATP than the NADPH. Because we know that from the non-cyclic photophosphorylation, it's able to produce two types of product. One is ATP, and another one is the high energy molecule, which is NADPH. But for certain circumstances, the plants they need more ATP. So to produce more ATP, the plants will switch to cyclic photophosphorylation, because it is a shorter pathway, so they can produce ATP in higher rate. So when the ATP amount is adequate or is sufficient for the processes, it will switch back to non-cyclic photophosphorylation. Both non-cyclic and cyclic electron flow are types of light-dependent reaction. Dependence so means that they require light energy or photon. So it will occur at the thylakoid membrane of the chloroplast, because here is the occurrence site of chlorophyll or the photosynthetic pigment. They able to absorb light energy. So the main function of the light energy absorption is to produce the ATP through a process photophosphorylation. But due to the difference in the route of electron flow, iaitu dia punya perjalanan pergerakan electrons itu, the products are different. The non-cyclic electron flow will produce ATP and the high energy molecule and ATPH, or can call as electron carrier. And the cyclic electron flow only produce ATP. This is closely related with the photosystem involvement. For non-cyclic electron flow, they involve both PS2 and PS1, and cyclic electron flow involve PS1 only. And for non-cyclic electron flow, the water is actually the first electron donor, because the electron deficiency occurs in the PS2, which is the primary pigment chlorophyll A P680. The lost photoexcited electron will be replaced by photolysis of water. However, in cyclic electron flow, the electron donor is PS1, or the primary pigments found in the reaction center of PS1, which is chlorophyll A P700. And in the non-cyclic electron flow, NADP+ is the last electron acceptor. It will accept the 2 electrons from the ferredoxin, the second ETC, in the non-cyclic electron flow, and combine with the proton from stroma and produce NADPH. However, in cyclic electron flow, PS1 is not only the electron donor, but itself is the last electron acceptor. This is why we say the electron flow is cyclic, di dalam keadaan kita semula. And the non-cyclic electron flow involve photolysis of water, and they able to produce the oxygen as byproduct. However, in cyclic electron flow, photolysis of water do not occur, so no oxygen is released.